All right, let's dig in. The four forces of evolution. I created that background myself. It's pretty cool, right? Four forces of evolution. It sounds like Harry Potter and the four forces of evolution. Okay, so a force of evolution is something that can essentially change the frequency of alleles in a population. So a fancy way to say that is it can push genotypes and allele frequencies out of equilibrium in a population. So equilibrium just means like stasis, that if there's a certain frequency of eye colors in a population, equilibrium would mean that generation after generation those frequencies stay the same. So a force of evolution is something that can change that equilibrium. Okay. Recombination is not a force of evolution. Remember, recombination is sort of the shuffling of the deck during meiosis, so that your chromosomes sort of shuffle up because you can't pass on 100% of your DNA to your children. You only pass on 50%, and which 50% is the question. So that's where the shuffling comes in. But that doesn't create new variation, and that doesn't that in and of itself won't change the average kind of frequency of a trait in a population, because it's random, and over time those fluctuations of what's getting passed on should even out. It doesn't create any new variation. It just shuffles the existing variation in individuals. So if that's not a force of evolution, what are the forces of evolution? Well, the first one we've already talked about, natural selection, where based on the environment, and that is key. I don't think I emphasized that enough in the last video, but the fitness of a trait depends on the environment. In an environment in which dairying is not necessary and is not practiced, the frequency of lactase persistence is low, meaning the frequency of lactose intolerance is high, because milk and dairy products are not necessary and they're not consumed. So, however, in an environment in which other forms of sustenance aren't enough, and in which it makes sense to have large herds of dairying animals, then a mutation that allows you to consume milk products and absorb all their nutrients into adulthood would be beneficial, and it would rise in frequency in those environments. And just like the peppered moth, in the original environment, the light-colored moth was more fit. Again, it's a relative to the darker-colored moth. But as the tree trunks became darker, the environment changed, then the trait that was fit changed, and it changed to the darker one. It was more fit relative to the lighter one. Okay, so natural selection can change frequency of alleles in a population. Mutation can also do that. By itself, it doesn't do it a whole lot, because you're not going to have a bunch of spontaneous mutations that are all the same. Like, everybody's not going to spontaneously mutate to have purple eyes, right? There might be one person that happens in, or two people, but that's not something that's going to spontaneously happen to a bunch of individuals. So it's not a real strong force by itself, but it's a necessary force, because that's how new variation is created, is through mutation. Genetic drift is complicated. That's going to take some more explanation. But genetic drift is essentially random fluctuations in allele frequency. And genetic drift is a stronger force in smaller populations. And I'll explain why that is later. And then gene flow. So within a given population, you've got a certain gene pool, a term you're kind of familiar with, the variations of alleles that exist within that population. But you can get individuals coming in from other populations. You can get sort of migration into a population, and that's a form of gene flow where new variants or different proportions of alleles are mixing in with that population. And it can also work the opposite way, where you have variation draining out of a gene pool. So that's gene flow. So we'll go over the three, aside from natural selection, in a little more detail. There are many types of mutations I already mentioned, like SNPs, single nucleotide polymorphisms. They're also sometimes called point mutations because they happen at a specific point. Insertions and deletions we already talked about. What are the causes of mutations? How do these things occur? Well, through DNA copying error. So anytime your DNA is replicating, making another copy, either in mitosis or meiosis, it's just like, imagine if you were hand copying Shakespeare. If you were hand copying an entire work of Shakespeare, you would be extremely, extremely lucky if you didn't make any mistakes whatsoever. And the rate at which this happens is quite low, but it happens. Now, a mutation that occurs during mitosis cannot be passed on to offspring because, let's say I spend a lot of time in the sun, I get a mutation in the skin cell, and that mutation, maybe leads to skin cancer eventually. I can't pass that specific thing on to my children because what I pass on to my children comes from sperm or eggs. So only mutations that happen in the germline cells, the sex cells that produce gametes, sperm and eggs, only those mutations can be passed on. Now there are some things that can be passed on that may increase your likelihood of getting skin cancer. For example, light skin tone, right? That's something that can be inherited, and that increases your risk. But getting skin cancer in a particular location is not something that gets passed on. So also, as I was just indicating, DNA damage from chemicals or radiation that can cause mutations, right? So either the copying when you're replicating DNA, or let's say you get incoming UV radiation, it damages the DNA in one of your cells, and when that DNA repairs, it doesn't repair properly, and so that can cause a mutation. It happens quite rarely. I saw one estimate is about 1.1 times 10 to the negative 8. So 0.00000011 per. Basically, that's the frequency. So per haploid genome means like a human genome essentially. So a haploid genome has about 3 billion base pairs. I also saw somewhere else where there's about somewhere greater than about 60 mutations can be inherited per newborn on average. Genetic drift. Genetic drift I'm going to go into even in more detail in I think the next slide, but it's like I said, it's random allele frequency changes. It affects smaller populations more powerfully because of the size of those populations, which I'll explain a little bit more in the next slide. There are a couple of subtypes of genetic drift that get special names because they are important in the study of population genetics. One is founder effect, and the other is genetic bottleneck, and they're quite similar. Founder effect is where you're taking a subsample of a larger population, and that subsample then goes on to found its own population. And you know what, let me actually, if we were in class, I would do this experiment. I did this experiment in some of my other classes, and these slides come from that experiment where I got a couple big bags of M&Ms, and I actually separated out the colors and counted them. I did that. Yeah, I apparently have nothing better to do with my time. And in one case, this was the distribution I got. There were probably 700 some odd M&Ms that I counted or more, and there were a little over 100 yellow, somewhere around 60 brown, 90 blue, about 145 green, about 160 orange, and about, I don't know, 45 red. And so these are the frequencies in this pie graph in the giant bag of M&Ms that I got in the parent population, right? So, orange and green were at the highest frequencies, and red and brown were at the lowest frequencies. And then I had students take small samples of those, not with their hands, of course. I had little cups. Take small samples out of the bag, small samples, right? So in those small samples, you're unlikely to get the exact frequency of those colors as they were in the parent population, right? It's random. You shake the bag up, you take a little cup, you might not get exactly 27% orange and 23% green. And the smaller your sample size, the more likely you are to deviate from that. So imagine you took one M&M. Well, then you're going to get 100% of one color and 0% of any other colors, right? Imagine if you take 3 M&Ms. Then, you could get 100% orange, you could get 100% brown, or you could get 33% orange, 33% brown, 33% green, and 0% every other color, right? So these were the examples in this particular class that the slide came from, where I had 4 different students do it, and this is the original parent population, right? So you'll see in sample 1, orange was 40%, so that differs from the 27%, right? I don't know how many M&Ms this student had, maybe it was 20 or something. And then here, there you see the 27%, okay? But red varied from 5% at the lowest to 10% at the highest. Let's see, blue, actually, it went from 15% in the parent population to a low of 7% in one sample and a high of 20% in another sample, okay? So I'm just telling you something that you know intuitively from experience, but maybe you don't think about on a regular basis. And so you can imagine those little subsamples of M&Ms as being founder populations from the parent population of the giant bag of M&Ms. And so a founder population can differ significantly in the frequency of traits compared to the parent population. And then when that founder population then, kind of becomes its own gene pool, and all mating is occurring within that population, over generations, you can get a population down the line that is significantly different from the parent population. So here's just another graphic of that, where you've got the original population of these ladybugs. You've got some dark ones, you've got some lighter ones with spots, you've got some gray ones with spots, you've got some dark spots, some light spots. And then let's say you take a subsample of that, and that becomes a founding population. And in that subsample, you have 2 black ones and 2 lighter ones with spots. And then the descendants of that population, look like this, where the frequency of the black ones and the lighter ones with spots is a lot greater than it was in the original population. But you could take a different founding population, randomly chosen, that just happens to have only the gray and the light colored ones, and the descendants are going to look different than the other founding population, and they're also going to look different than the original parent population, right? So founder effect is important because in any animal populations, but since we're in anthropology, we're talking about humans, it happens a lot. A couple examples that I can think of off the bat are the population of modern day French Canadians. Originally came from a relatively small founding population from mostly from a particular region of France. So France was the parent population. It was only a small subset of people there that went to Canada for fur trapping. And there are some differences in frequencies of traits versus the original population. Not all these differences are going to be harmful, but the examples that I know of are disease related because those are the ones that get reported more. But for example, French Canadians have a higher frequency of I think it might be Tay-Sachs disease than the original French population or the modern day French population. I think it's Tay-Sachs disease. The Amish are another example where they came. The founding populations that came to the United States came mostly, I believe, from Austria, maybe parts of southern Germany, and there was a relatively small founding population that came to the U.S. And of course, they're very endogamous, meaning they marry only within their group. They don't really bring in outsiders, and so. They happen to have a higher frequency of polydactyly, so like 6 fingers or 6 toes, compared to the populations that exist in Austria and southern Germany. Just because maybe they got, 1 or 2 more individuals in those founding populations that happen to have that trait. So it spread to higher frequency in the founding populations. So this, these founding populations, this founder effect occurs a lot in human societies. Thought there was another example I was going to give, but the other one is genetic bottleneck. And a bottleneck is just where it's kind of what it sounds like. Imagine you've got a bottle filled with colored beads and you tip it upside down. Well, there's a narrowing of the neck, and so only certain ones can come out, and it's random, of course, which colors come out, as long as they're randomly distributed in the bottle, and that population that makes it through the bottleneck is obviously smaller, so it's carrying a smaller amount of the overall diversity, genetic diversity of the rest of the bottle. So how does this apply to human populations? So if there's a natural disaster or a famine or some kind of cataclysmic event, like Pompeii or something, the population is going to be reduced in size, and it's also going to be reduced in its overall genetic variation. So you're not going to get the same proportions of alleles in the population that makes it through the bottleneck. Some may not be there at all, just randomly. So, and that's also happened a lot in human history, because our population of Homo sapiens was rather small early in our history as a species, and there have been at least a couple events where, because of ecological, environmental, climatic changes, the kind of ecosystems that our species was, most adapted to survive in became, smaller and smaller areas. There was probably famine, competition for resources, and so the population shrank, and so there have been many bottleneck events in human history. I mean, even like the Black Death in Europe killed a huge proportion of that population. So that's even kind of a little bit of a bottleneck effect. I'll get back to why genetic drift impacts small populations in particular in just a little while. But gene flow is much simpler. It's genes exchanged between previously isolated populations, new proportions of alleles coming in, or new novel alleles coming into a population from the outside. And that's happened a number of times as well. So if you think about the Columbian exchange, or the European colonization and conquest of the Americas, obviously you had populations who had been separated for a significant amount of time, populations in the Americas and populations in Eurasia. And then all of a sudden you have these mixing of populations, and that's a type of gene flow. And so bringing in different variants, one way or the other, it obviously can change the frequency of alleles. So that's one of the forces of evolution. Let me. I'm going to discard my ink annotations. Let me show you this cool spreadsheet that I developed. TM. It's about a coin flip. Now, okay, here I have a fair coin. I promise. It's a quarter, heads and tails. And, if I ask you, what's the probability that I'll flip it and I'll get heads, you'd say, 0.5, 50%. If I flip it 10 times, how many times do you think I would get heads? And you'd say 5. But if I asked you, what if I got 4 heads? Would you think that was crazy? You'd be like, no. What if I got 6 heads? It's not crazy, right? 5 is the average. 5 is what you expect. That's your expectation value, because each time you flip it, there's a 50% chance you're going to get tails, a 50% chance you're going to get heads. So what if I flip it 4 times and I get 4 heads? Are you going to think there's something magic going on, or I'm cheating? No, it's totally possible to flip it 4 times and get 4 heads. It's not that crazy. What if I flip it 100 times and I get 100 heads? Well, then you'd start to think there is something going on. And that's the difference between small populations or small sample sizes and large sample sizes. In large sample sizes, the randomness of the flip, it will tend to balance out. You might get 4 heads in a row, but then you'll start to get, more tails in a row at some point. It'll even itself out eventually, right? Because if it's true that it's about 50-50 for each flip, then the greater the number of flips, the more those random events will balance out. And the greater the number of flips, the less each additional flip impacts the total proportion, right? So let's say I have 50 heads and 50 tails in 100 flips, and then I flip it 101 and I get a head. Well, that doesn't change the proportion that much. It becomes 51-50, right? But if I flip it 4 times and I have 2 heads and 2 tails, and then I flip it a 5th time and I get a head, all of a sudden it goes from 50% heads to 60% heads, if I did my math in my head right. Okay, so let's just demonstrate this. This might be boring. I might regret doing it, but I'm gonna flip it. So flip 0. I have 0 heads and 0 tails. I'm only gonna mark the heads because that's the way I created this. So flip 1, I got tails. I'm not gonna show it to you each time. You're gonna have to trust me. Okay, so all right, so I'm not gonna write anything. I'm gonna move on to flip 2. So flip 2, I get tails. All right, I'm gonna move on to flip 3. Flip 3, I get tails. Oh my God. Flip 4, I get heads. All right, so you see that it's rising in frequency here from 0% up to 25%, so 1 out of 4. Oh, I got heads again. That one's not good. That one's not good. I got heads again. I promise you I'm not faking this. I got heads again. I'm obviously not a great flipper. That time I got tails. That time I got tails. I don't know if I'm going to do all 20 because I'm sure you're bored to tears. I got tails again. This doesn't seem very random. Tails again. Heads, finally. Heads on the 12th. Heads again. Heads again. Isn't this riveting? Tails. Yeah, I'll go all 20. Why not? Tails. Tails. Wow, this is so weird. I wish I had some other people flipping it. Heads. And one more? No, two more. Tails. Ugh. Tails. All right, that one kind of sucked, but all right. So you see, the largest changes in frequency occurred early on, and then later the changes in frequency are getting smaller, and eventually, if I flip this, 100 times, it should start settling in, around the expectation value. Okay, so in small sample sizes, you can get wild deviations from your expected frequency, but in large sample sizes, you're only going to get small variations from your expected frequency. And that's why genetic drift impacts small populations more, because some kind of randomness has a big effect. So imagine that there's a population of 50 individuals, and there's 2 individuals with blue eyes, and there's some kind of cataclysmic event, and it affects individuals randomly, and now both of those individuals with blue eyes just happen to die in this cataclysmic event, and so now there's no one to pass on that trait. In my hypothetical example, because your frequencies are impacted more by events in small populations, they change the outcome of the future events more. Okay, here's another example. Sticking with eye color just because it's an easy thing to talk about. I have grandparents on both sides of my family that had blue eyes. And my parents had 4 kids. I'm one of 4. And so there was, I don't know exactly what the probability was, but I think it was about 50-50 that maybe we could have had blue eyes. Each one of us had a 50-50 chance. And guess what? Out of 4 of us, there are none of us that have blue eyes. And let's just imagine that we're not carriers either. We probably are. Some of us probably are anyway. But, now, just through happenstance, maybe we will not pass on that trait to any of our children, and all of a sudden it's gone. Or maybe only one of us carries the trait, carries an allele for that trait. And maybe it'll get passed on, but it'll get passed on at much lower frequency. And that's just random. That's because my parents had 4 kids, not 25, right? And so it just happened that when they flipped the coin, they got heads 4 times instead of 2 heads and 2 tails. Okay, I hope that makes some sense. So I'm going to go back to the PowerPoint. So selection versus drift. Selection is most effective when population sizes are large. Number 1, that's because drift is stronger in smaller populations. But number 2, it's because selection feeds on variety. So you have to have variation in order for selection to work, right? If there were no variations in size of wolves or dogs, then you wouldn't be able to get a Chihuahua. If there were no variations in the colors of those peppered moths and the environment changed, well, the population couldn't change, so selection couldn't work because there was nothing to select from. Okay, so the larger the population, the more variation you're going to have. You don't need to know that. Okay, drift. You don't need to know this stuff. Don't worry about this. We don't need to worry about that. Drift is most powerful in small populations, as I said before. Okay, so there's sort of this push and pull then. All these forces of evolution are occurring at the same time, and all populations are evolving. It's impossible for living populations to not evolve at all. There's a difference between evolution and speciation. Speciation happens over long time scales, and you need some other factors in there, but evolution is not synonymous with speciation. Speciation occurs through processes of evolution, but when I'm talking about evolution, I'm talking about a change in allele frequency in a population over time. And so allele frequencies are always changing, either through these little random occurrences, like none of my brothers or I having blue eyes, which is just based on the shuffle, right? Based on what gets passed on, it's random. Or based on selection, or based on mutation, or based on gene flow. Living populations are always evolving. And so the way that they evolve is based on a balance of these forces of evolution. So there was a early geneticist, Sewell Wright, who came up with this idea of an adaptive landscape. And it's essentially the idea that within this, landscape, that's a landscape of fitness. So how fit a certain combination of traits is. So a combination of traits based on gene 1 and gene 2, and the height is fitness, right? So this dimension is fitness relative, Within this landscape, that is the maximum fitness in that landscape, right? But selection can only push you up towards greater fitness, right? So let's say the population starts with the average fitness being somewhere here. Selection will move you up to like maybe some kind of local maximum, but that's not a global maximum in this environment. That's not as high as it could be, right? But selection can't take you from this peak down to get to this peak, because selection only increases fitness. So Sewell Wright said, you know what, maybe genetic drift is an important component even though it's random, it might be an important component in helping selection to continue to increase fitness, because only something like drift could push you off of this peak and down into an area where then selection could push you up again to another local maximum. All right, so that's just something kind of cool, interesting to think about. But what I want you to take away from it is simply that selection is more efficient in large populations, drift is more efficient in small populations. They're both occurring simultaneously in all populations, as long as there's variation, and some traits are more fit than others in particular environments. And I also would like you to know that not all, we assume that if we exist today as we are, then that must mean that we are maximally adapted. There's nowhere to go. You can't get more fit, but that's just not true. There's never such a situation. Number one, it depends on the environment. Number two, not all forces of evolution are about adaptation. Genetic drift has nothing to do with adaptation. Genetic drift is random. Only selection has to do with adaptation to a particular environment. If the environment changes, what is the most fit in that environment also changes. So think about that for a little while. That's it for genetics and evolution. I hope it wasn't too mind-blowing. If you have questions, you know where to reach me. Enjoy the rest of your day, week, however long until we speak again. I'll see you soon. Bye-bye.